Hi everyone, it's Miss Lopez. This is part 2 of the phylogenetic trees activity. Make sure you've watched part 1 before you move on to part 2. One way to discover how groups of organisms are related to each other is to compare the anatomical structures, so body, organs, parts, of many different organisms. When different organisms share a large number of similar structures, it is considered strong evidence they are related to each other. If there are specific modifications of those features shared by different groups of organisms, we say that these features are shared derived characters. The following is a phylogenetic tree for mammalian structures. So you can see here where the structures for mammals are considered chordates. So you can see some of the characteristics that mammals have in common with other animals on this phylogenetic tree. So question 10 asks that, gives you instructions that the common ancestor for all animals is believed to be an organism called a choanoflagellate. Circle the choanoflagellate in red. So that single-celled ancestor that's at the root of this phylogenetic tree is the choanoflagellate. Number 11. What are 3 physical characteristics we can use to create phylogenetic trees of animals in the tree above? So you're going to look at any of the derived characteristics such as multicellularity, the arisal of tissues, the 3 germ layers and bilateral symmetry, the development of the protostome, the coelom, the deuterostome development. Any of those can be used, or even radial symmetry, pseudocolum, radial symmetry again. Any of these are going to be acceptable to use as a characteristic to create these phylogenetic trees. So in order to fill in the table, we are going to look at echinoderms and chordates, and we're going to try to identify what they have in common. Let's find them on the phylogenetic tree first. So here are the echinoderms, which are the spiny-skinned, that's what the name means, echinoderm, spiny-skinned animals, and right next to it are the chordates. And we're going to look to see which which trait they have in common. You'll notice here that echinoderms have radial symmetry, and that characteristic is placed on a branch separate from the chordates. So that means that radial symmetry is unique to those echinoderms. We're going to have to go a little bit further back to see what they have in common, and you can see right here that what they have in common is the development of the deuterostome. The next part of your table asks you to list 2 organisms that evolved prior to a coelom. So first we're going to have to find where the coelom is located on the tree, and then we're going to have to identify some organisms. So right here is the arisal of the coelom in this tree. So the way that this works is that the common ancestor, that choanoflagellate, is the oldest, whereas on this end of the tree, your chordates, including humans, are more recently evolved. So once you have identified where that coelom is, you can take any organism prior to that, in other words, to the left of the coelom on the tree. So you can list roundworms, you can list flatworms, you can list the cnidarians, or you can list sponges, which is the genus Porifera. The next column in your table asks what are the closest relatives to the annelids. So the annelids are a type of worm. They include like earthworms, for example, and they're located right here on the table. So when you want to find the organisms that are closely related to the annelids, you're going to have to find the ones that are similar on, or at least close by on the tree. So annelids have a branch here, and so right here is a common ancestor between annelids and their closest relative, which is the arthropods. The other organism that's going to be common to the annelids is still going to have a recent common ancestor or a recent node. And so that's going to include this point right here, which would include your group of called the mollusks. The last question on your table is now asking you, instead of comparing the organisms themselves, it's going to ask you to identify the specific trait, the derived trait that they have in common. So it's asking for the cnidarians and the mollusks and asking what trait do these two have in common. So what you've got to remember in a case like this is that any trait that you see to the right of this branch is not found in the cnidarians, which are the ones that have stinging cells. So for example, the coelom, the pseudocoelom, the protostome, the 3 germ layers, all of these are not good candidates because they are not present in the cnidarian. Now, the cnidarian does have radial symmetry, but you can see that this is something unique to this branch. You can see how the radial symmetry here doesn't extend at this point. So even though cnidarians have radial symmetry is not in the mollusks. So the only thing, there's 2 things that mollusks and cnidarians are going to have in common. That is going to be tissues and multicellularity. Thank you guys so much for watching part 2 of your phylogenetic tree activity. I hope this video was helpful.